Welcome, everybody, for today's seminar. We have the pleasure of having Christopher LaRock. He is an associate professor in the Department of Microbiology and Immunology here at the School of Medicine. His lab examines how microbial biovirulence factors, such as toxins and effectors, and host factors, such as cytokines, interact using biochemical, genetic, and pharmacologic tools. His work is revealing new mechanisms of microbial pathogenesis, infection risk associated with immunotherapy, and new drugs targeting infectious and anti-inflammatory diseases. Without any further ado, please welcome Dr. LaRock.
Thank you all for having me here today. And so I'm going to talk about bacterial determinants of morbidity, mortality, and epidemics, comparisons between plague and scarlet fever. And I've really enjoyed the seminar series that I've taught so far this year. And in the spirit of that, I'm going to give a slightly different or substantially different talk than my usual one, because I think it's important to introduce a sort of framework in which my field conceptualizes our research and techniques we use. So it's not going to be as data heavy as it is going to be concept heavy. And so I'm very interested in how pathogens cause disease. This is my lab here over the years, and I'm also going to acknowledge our funding sources. These are the people that did the experiments that I'm going to talk about today, and then the people that paid for those also. And we have some learning objectives I'd like to accomplish today. First, to define what is a pathogen. How do we determine what is a pathogen versus what is not? How to discover what makes a pathogen a pathogen. And this is usually accomplished by specific things that accessories of the microbe that it uses to interact with its host and manipulate it for its own benefit. How to avoid sloppy thinking and other traps, which are important in all things, but in particular in the research topics I'll be talking about. Be less anthropomorphic, because as we dig into how these bacteria work, many of their strategies are very counterintuitive, and they're not things that you or I would choose to do, but it works very well for them. To embrace biological diversity, because each of these different pathogens I'll talk today have very different strategies and go about infection in completely different ways, but it still works for them. And evolution is always the cause for all of these different things. And so, what is an infectious disease, right? So germ theory states that some diseases are caused by microbes, and Robert Koch set out procedures for us to define what is an infectious disease. And as per his original postulates, I'm sure you all know these. I'll recap them very briefly, though. That the agent should be present in a diseased organism and absent and healthy. It should be able to be isolated and grown, cause disease when introduced into a healthy host, and re-isolated from the experimental host. And this works pretty well for a lot of circumstances, but not all, right? So you can imagine, especially with the microbiome, but the Helicobacter is a great example of this, right? So some strains, but not all strains of Helicobacter pylori will cause stomach ulcers, which can lead to stomach cancer. And so both healthy people and diseased people will have Helicobacter, but the healthy ones might just have strains that aren't capable of causing those ulcers, right? And then, for isolated and grown, that falls apart with, some viruses and prions and diseases like this. And then in microbiome and microbiota-mediated diseases, it can be a dysbiosis, such as overexpansion of Clostridium difficile, which is present in healthy people and completely harmless, right? But when it takes over your microbiome and takes over your flora and makes too many toxins, that's when it causes a disease state. So it captures a lot of disease, but not all of it. And once we define, what a pathogen is, we have to define how it's causing disease, what specific factors it does. And we have a molecular form of Koch's postulates that was introduced a number of years ago by Stanley Falco. And this is kind of the bread and butter of what we do in my lab, is we look at a pathogen and a phenotype or a gene should be present in that pathogen and absent in closely related non-pathogenic microbes. And most serious pathogens do have closely related species that aren't pathogenic toward humans. And so you can do that by genomic analysis, which is quite easy nowadays. And then if you delete that gene through genetic manipulation, for example, inactivation of that gene should lead to a loss in pathogenicity, right? So you're taking away something that bacterium needs to survive in its host. And then if you bring it back, then you should get a restoration. So that shows that your mutation of the gene is specific to exactly that mutation you introduced and not any collateral genomic damage, for example. And then not one of his original points, but one that I put on here because it really speaks to a lot of the experiments that my lab does, is that there'll be a reversal of attenuation in a modified host. So if you take away a virulence factor of a microbe and render it dead when you put it into a host, if you then also manipulate the host and delete whatever the target of that virulence factor is, then that previously attenuated, that previously dead strain should be able to grow in that compromised host. Right, so again, this is the basic experiment. We can do genome gazing, and we can look at 100 genomes of salmonella, and all of them might have this one thing that we think is a virulence gene. And if we look at a closely related enterobacteria, you see that's not pathogenic. It doesn't have a gene that looks like that. And if we put it into a host organism, like a rabbit, it'll kill it. And if we genetically manipulate the bacteria to get rid of that gene and put the same amount into a host, that host survives. So my lab, we never do survival experiments. We typically do more nuanced things rather than just live or dead, which is easy to do. It's binary, but it's not necessary to cause that level of mortality. And so we typically look at more specific aspects of disease, such as if we're modeling strep throat, we see if there's tonsils getting bigger, or we'll look at specific cell populations and how they're changing in response to bacterial factors and things like that. And so there's a quote that I want to bring in here before we move on to this next stage from Antoine de Saint-Exupéry, who is more famous for writing The Little Prince in this book, but this book is quite good. I think most grad students would like it. So he was an early airline pilot in the Postal Service, delivering mail, and he was flying over the Sahara and crashed, and he thought he was going to die out there. And so this is about his musings on life and certain death and despair, and which is a lot like grad school. That's kind of the parallel I'm trying to make. But the purpose of this quote: so protection is finally attained not when there is no longer anything to add, but when there's no longer anything to take away, when the body has been stripped down to its nakedness. And so I think it's useful to think of this idea when we're thinking about bacterial genomes, because we often think of more is better, right? An iPhone 10 is better than an iPhone 1 because it has 5 cameras instead of 1, right? And having something extra just in case you need it is useful. But bacteria don't work that way, right? So there's mutations happening every generation. They're constantly losing stuff if there's not selection to maintain it. So they don't hold on to their garbage. They are slimmed down to mostly just what they need. And so when we do these sorts of experiments where we're deleting a gene, I would expect almost all the time where we delete a gene in a microbe, that microbe is going to be less virulent, right? And it doesn't necessarily tell us what we deleted is a virulence factor or is important in pathogenesis. It could just be a tRNA or something that the bacteria needs to metabolize glucose. It could be completely irrelevant for the question on how the microbe causes diseases. And so the parallel, bringing back to that quote I had, the parallel is to studying how planes fly by taking away pieces, right? You could have 1,000 planes or 1,000,000 planes, and in each one you delete a different component, right? And airlines or airplanes are very stripped down, right? You've sat in airline seats, right? They get rid of every spare ounce they can because more weight is more fuel. And if you go into the cockpit, there's lots of dials and knobs, and I assume everything is there for a reason, right? And so you study how planes fly by taking things away, probably a lot of the planes will be, fine most of the time, for some distance, right? Because you're getting rid of a seat or something like that. A lot of planes will just never work at all because it won't have landing gear, so it can't get off the ground, or it'll spin in circles because you're missing, whatever. But that's not the question you're asking by understanding how a plane flies, right? You want to understand, lift, how the plane is bending the air around the wing over the wings in order to gain lift, and you want to understand how the plane is getting thrust, so it's moving forward, right? So, those are the parts of the plane you want to study, not all the rest of the stuff that is maybe, essential for the plane to fly, but it's not telling you how a plane flies. Does that make sense? And so that's what we're trying to get at with the bacteria. Everything is important in the bacteria, right? I'm not saying anything is not important, but I'm only interested in the parts that are causing disease. And so our first example is going to be plague. And I chose this example first because it's a relatively simple one as far as this concept goes. So plague is an important disease. Historically, it's killed a lot of people. It is endemic throughout most of the world in wild rodent populations, and it kills a couple of people in the US every year, but it's not a major source of morbidity and mortality in the modern age. And it has a fairly complicated life cycle. So in the wild, it's transmitted by a flea vector between wild rodents, such as ground squirrels and mice and marmosets. And then it can also be transmitted through a pneumonic route. In humans, when they get it, it's often, it can be either pneumonic or from the flea vector from a wild animal. In humans, then it can be, historically, it could have been transmitted by human louse, but humans don't commonly have that anymore. And then in these big plague epidemics, it would probably also went a pneumonic route. But I'm going to focus on the flea here, because this is what plague has evolved to do. So everything it does has evolved to do this, and it just happens that it can work in humans and it can work by a pneumonic route also. And so the transmission goes that a flea feeds on a septic mammal. It intakes Yersinia pestis with the blood meal. Later, so initially colonizes the midgut of the flea, and then later the proventriculus, which is this valve here. And as it's growing there, it forms a biofilm. So this mass of bacteria that's embedded in a sticky matrix of carbohydrates. And then as the flea is attempting to feed, it regurgitates the Yersinia pestis into the mammal. And so there's several things going on here that we have to molecularly study to understand how this works. And so, for example, this biofilm on the proventriculus, what that does is it blocks feeding of the flea to make it hungry, and so it bites the host more often, and so plague has more chances in order to be delivered into a target mammal. And then also this biofilm flakes off in chunks, and once it's delivered into the mammal, this provides initial protection against the immune effectors of the host, because free living bacteria are quite readily killed, where this globby mess of bacteria is less readily killed. But the other challenge I want to highlight is this initial one right up here. So the flea has to feed on a septic mammal. And this is the really hard part that I wanted to understand in this next experiment. So humans, you can develop a bacteremia, which is bacteria growing in your blood, and that can go on to sepsis, which is fatal, or which can be fatal, and that's a overwhelming immune response to the bacteria in your blood or in your body. Bacteremia rarely kills people. It's usually sepsis that kills people. It's the immune response to the infection that kills you. And so the infectious dose of Yersinia pestis for a flea is quite high. It's on the order of 10 to the 1,000 or 10,000 or higher CFU going into a flea. And the flea, as you can imagine, is feeding on a tiny volume, 1 microliter or less. So the mammal has to have a very high degree of sepsis in order for a flea to pick up the Yersinia pestis, right? And it's a high enough burden that any other bacteria, the mammal would be dead. And so in order to, for pestis to transmit, it has to immunosuppress the host enough to have bacteremia without sepsis. Because now also the important part about the flea biology is a flea won't bite a dead human or a dead mammal, right? So they bite warm-blooded mammals, and also if a person is dead and their blood is coagulated, it can't feed. So so plague has to keep a person, you know, really kind of in a zombie state with a huge bacteremia, still warm-blooded, not coagulated in order for this transmission. And I wanted to know how it's accomplishing that. And so for that, going back to this molecular postulates, I'm going to skip some biochemical stuff, but I discovered that Yersinia pestis makes this virulence factor called YAP-M, which is this immunosuppressant factor. And without going into the immunology, what it does is it inhibits this protease called caspase-1, which is a key regulator of the immune response. And this is completely essential for Yersinia. So if we do an experimental infection of mice with Yersinia pestis, or in this case, it's a different Yersinia as a surrogate for pestis, but it's true for pestis also. If we do a sepsis model and we infect mice with wild-type Yersinia, it grows quite well. We took the spleen because it's a blood-rich organ, and it's easier to collect more reproducibly than blood. And this YAP-M toxin is essential because if we take it away, it doesn't grow as well. And then we did this last experiment, and then you know, other experiments I'm not showing. We did complementation and all the rest of these postulates. But in this very last one, attenuation, there's attenuation reversal on a modified host. So if the major thing important about this toxin is that it inhibits caspase-1, and that's how Yersinia gets bacteremia without sepsis, then if we take away caspase-1, then Yersinia shouldn't need YAP-M anymore. And that's exactly what we saw, that we had reversal of the attenuation of the YAP-M mutant when we put it in these genetically immunosusceptible mice. And so that's one of the more simple examples. Moving on to a more complicated one is group A streptococcus. And there's many reasons why it's more complicated. One is that it has a lot more different virulence factors, a lot more known ones, and probably a lot more unknown ones also. Many of them are multifunctional. So one virulence factor might have multiple host targets. Also, multiple virulence factors might inhibit the same host target. So there's a lot of functional redundancy there. The consequence of this is that group A strep is very resistant to our immune system. So, and that's the basic basis of a classic experiment called the Lansfield experiment, named after Rebecca Lansfield, who back in the 1930s found that you could take human blood and you spike it with your different pathogens of interest, and group A strep will grow logarithmically right from the start, where other bacteria, some of them will survive, but they at least have a refractory period. So the basis for that is because group A has so many different toxins to resist everything that the blood can throw at it. The other special thing about group A strep is that it causes many different diseases. So Yersinia pestis causes plague, just like Vibrio cholera causes cholera, and Mycobacterium tuberculosis causes tuberculosis. But group A causes more diseases than any other species of microbe. You're all familiar with streptodenture pharyngitis, and this is the most common group A infection in North America and Europe. And so you get swollen tonsils. It's usually self-limiting. People often take antibiotics, and it works quite well. There's scarlet fever, which is a very similar sort of infection, but it's from strains of group A that have gained additional toxins that cause a more inflammatory infection. And so scarlet fever is you get a rash disseminating from your throat from all this excessive inflammation, and it's much more contagious than normal strep throat because this inflammation, as I'll show later, can drive transmission. Infections more important throughout other parts of the world are skin infections such as impetigo, which are more common in developing countries. And then severe infections such as necrotizing fasciitis, which are these flesh-eating infections, which I'll show you some models of also. And so here's an example of something that did go relatively simple in group A, where we had found this virulence factor called M protein is protected against an antibiotic of the immune system called capsaicin. And this is basically the same as that Yersinia experiment, where we showed that this factor M protein was essential for virulence. So we did this is a necrotizing fasciitis skin infection model of mice. And if we delete this gene out of group A strep, the bacteria with that mutation aren't able to grow very well in the skin. And again, I didn't show it here, but if we can complement it back on a plasmid and do all that sort of thing to fulfill these molecular postulates. But then this last point is also true, that there is attenuation reversal in a modified host. So if we take mice and we knock out this immune antibiotic called cramp, these cramp knockout mice, the wild type bacteria do just as well in each mouse, because they have this M protein, and that's what it's important for. But the M protein mutant, which was attenuated in wild type mice, grows fine in the knockout mice. So that's saying that the major purpose, at least in this model, of M protein is to resist this immune antimicrobial, and that this immune antimicrobial could be quite good at killing group A strep, only it can't kill our wild type normal group A strep because it can completely resist it using this M protein here. And so, going back to this challenge, those were kind of simple examples where we could go back and forth between the microbe and host and really be sure that we were studying a propeller or a wing and not, a landing gear. Now, in this next example, we're going to look more at the immune system to see how we define what is a factor of the immune system rather than just something that a human or a mouse needs to live. And so you would imagine that a mammal is similar to a microbe, where we don't have necessarily a lot of junk. The things that you have there are things that you need, and you can develop a lot of different mutations that will make you sick, but they're not necessarily things that are important in immune defense. And so one thing that we think is quite important in immune defense is a cytokine called IL-1 beta. So what a cytokine is it's a way for cells of the body to signal to each other. And most of them are pretty specific, where only some cells will make that cytokine, and only some other cell types can detect that cytokine. So it's just one way for one immune cell to talk to another. This one called IL-1 beta is like shouting, because nearly all the cells of the body have a receptor for it. So when you get activation of the cytokine, you turn on all inflammation everywhere, basically. And we know from decades of studies from knockout mice that have this gene deleted, that they're more susceptible to infection. And the way these studies are typically done is that an immunology lab will develop a knockout mouse, and then that mouse will make its rounds to all the different microbiology labs studying all the different microbes. And they put their microbe in it and they compare it to a wild type mice and say, is this cytokine important for fighting disease or not? And 99 times out of 10 or out of 100, they're going to say that this cytokine is important, right? Because it, typically everything is somewhat important, and there's a lot of biases going into that too, right? There's a bias for publishing, for one, where if a person doesn't find a phenotype, those papers don't get published, or they don't trust their finding and then do it in a slightly different way, and then do find a way that there's a phenotype. Nonetheless, the state of the literature is that IL-1 is important in the defense against all pathogens, the exception being Yersinia pestis, which I showed in a previous paper. The thing about IL-1 being so pro-inflammatory is that it sometimes is accidentally turned on. And when it's turned on, it's really bad. for the human that it's turned on in. And it's a driver of inflammation in a lot of inflammatory diseases, including all of them listed here. But especially and relevant for this talk is rheumatoid arthritis. And it's such a major driver of these diseases that if you were able to inhibit IL-1, you get rid of much of the manifestation of the disease. And so, of course, since these diseases are very important in the Western world, compared to these, we do have drugs to inhibit IL-1, such as these, and they're very effective at treating these autoinflammatory diseases. But what we've done here is also a natural experiment, right? So we've created IL-1 knockout human beings, and we've released them into the wild where they're free to get all these different infections, right? So do these humans look like the mice do it in the lab? And so the result from 19 clinical trials I pulled together here is that these drugs are quite safe. That compared to a placebo control, they have similar mortality and serious adverse events associated with them. And there's an elevated incidence of infections, but not dramatically so compared to most other drugs. It's not the sort of phenotype that we would expect based on the experimental mouse infection models, where mice are dying left and right as soon as they see a microbe. And so I dug in a little further using adverse event reporting system data. And so this is a database that is very prone to false positives and false negatives and plenty of errors, but it was just a training data set to inform these next experiments. And I asked the question, which species of microbes were responsible for the infections happening in the people that were on IL-1 inhibitors? And the one that really stood out is group A Streptococcus or Streptococcus pyogenes here, where most other species were about the same or slightly elevated. This one had an over 300 times increased incidence of reporting in people taking IL-1 inhibitors versus people taking any other classes of drugs. And this was very specific to the sort of interaction I was looking for, is that people taking IL-1 inhibitors had the increased incidence of group A again compared to other bacteria. And it wasn't, broadly all infections caused by group A strep, but it was very strongly biased toward these necrotizing fasciitis skin infections. And this was also specific to the cytokine IL-1 and not just all other pro-inflammatory cytokines. IL-6, I will talk about today, but we found that it has kind of an intermediate phenotype because IL-6 is only important for fighting some strains of group A strep, but not all strains, where all of them, IL-1 is important for fighting all group A strep strains. And so, moving from this human data, we wanted to see how true it is going back to the mouse model. And this is, again, a necrotizing fasciitis model where we introduce group A strep into the skin of a mouse and then measure its growth. And in mice that were receiving this IL-1 inhibitor, they had a significantly greater burden of group A strep after this inoculation, saying that IL-1 was important for combating this infection. And we confirmed this genetically. So I took mice that had the IL-1 receptor knocked out. So IL-1 was still there, but it couldn't do anything. And that looked exactly the same as the drug treated. So arguing that the drug wasn't having any other effects other than inhibiting IL-1 signaling. And there's no additive effect of knocking out the IL-1 signaling pathway, and also genetically and also by drugs. Again, showing that everything was very specific. However, what we did, it was an additional control here where this protein caspase-1 is a key regulator of IL-1. And so a caspase-1 knockout should look exactly like the IL-1 knockout. Usually they're even worse because it regulates stuff other than IL-1. However, we found no difference at all there, which was troubling, but it led us into a completely new and exciting direction. And so there we found that it was a bacterial toxin called SPB that activated IL-1 in place of the normal host regulator, caspase-1. And so what caspase-1 does is it cleaves pro-IL-1 beta. IL-1 is synthesized as an inactive zymogen, and it needs a cleavage event in order for it to gain activity. And this bacterial toxin will do that same cleavage event and render it active. And so we mapped this out, and this is a diagram of what IL-1 beta might look like based on an AlphaFold model. And so the normal cleavage site is here. This is aspartic acid residue. This bacterial toxin cleaves here, so at a different site, but it still renders it active. And we mapped out how much of the cytokine needed to be removed in order for it to have signaling activity. And we did that by in vitro transcription translation, where we used primer walking to go down the gene sequence to synthesize lots of different truncated proteins, and then we measured the activity of these proteins. And we found that it was a pretty limited region of about 20 amino acids that was sufficient for activation, and potentially any protease that cleaves there could activate IL-1 beta. And since then, we found a number of microbes that activate IL-1 beta in this manner, and they're diverse species, including Pseudomonas aeruginosa, which is a gram-negative species, and Staphylococcus aureus, which is gram-positive like Streptococcus, but not that closely related. And we found this is true for some other cytokines too, such as IL-18, which is also regulated by proteolysis and can also be activated by SPB and other microbial proteases. And so we were wondering, in the case of necrotizing fasciitis, activating IL-1 and IL-18 is bad for the microbe because it induces an immune response that kills the microbe off. And we were wondering, why would it do that? And so group A is an oligogamic pathogen, right? So it's evolved specifically to infect a human, and it's found nowhere else in nature. So it should be an expert at doing that and not do missteps. And so we thought there could be a purpose behind it, and that it might happen at a different body site than during these necrotizing fasciitis infections, which are relatively rare, and they're not transmitted. They're not horizontally transmitted. So there's no real selection for the bacteria to do them. It's usually more of a mistake or a misstep of the bacteria when it does them. And we found it was very different in different infection models. So at the top here is our necrotizing fasciitis skin infection model. IL-1 inhibition makes the infection worse. There's more bacteria. But if we do a strep throat infection model, again in a mouse, we find that IL-1 inhibition is therapeutic. It kills off the bacteria if we get rid of IL-1. So it's the complete opposite of these severe skin infections. And then we also have a transmission model. And in this model, we found that IL-1 inhibition also prevented transmission, but it was largely because there's just no bacteria there anymore. And so what's going on? We suspected that that the microbiome might be playing a role, and this was based on some data we had already done, where we found that if mice were pre-treated with penicillin, and then they had time for that to flush from their systems, they were more susceptible to group A strep in this pharyngitis infection model. And that's what we saw here. So if we give mice penicillin and then infect them, there's a greater bacterial burden in the throat. And then if we give penicillin at the same time as the group A strep infection, there's no bacteria there because penicillin will kill a group A streptococcus. And so we knew the penicillin was likely disrupting the microbiota. And we knew that IL-1 was probably mediating inflammatory responses that were bad or that could affect group A strep. So we did this model again. Penicillin makes group A strep infection worse if it's administered before infection. And if we use this IL-1 inhibitor drug, then penicillin still makes the infection worse, but what's the point here? Oh, right. And so again, IL-1 inhibition means the bacteria can't colonize at all. But if we do that in combination with penicillin, we abrogate that and the bacteria can grow even if we inhibit IL-1 with anakinra. And it's the same thing if we ablate neutrophils, which are one of the major populations that are responding to this.
Yeah.
When you have strep throat, sorry, what?
Oh,
When you have strep throat, the white spots on your tonsils are
yeah,
white
coming,
that are responding to the infection.
coming. I'm on a Zoom. Oh, right.
And so again, so if you right, penicillin pre-treatment abrogates the need for neutrophils for these infections. So these data argue that the symptoms of strep throat, where you have a lot of inflammation and swelling, and the neutrophils, they're not an accident. That strep really needs them to survive in that niche. And it's probably in part for competition with the microbiome. So if you look at the upper respiratory microbiome, it's mostly Streptococcus. It's other species of Streptococcus. And when you have closely related species in the same niche, there's a lot of competition between them because they have similar metabolic pathways. And so in this experiment, we were trying to take advantage of that in order to better treat infections. And so Strep salivarius is closely related to group A strep, and it's commonly used as a probiotic strain. So it's generally recognized for safe in humans, and there's been a number of trials studying it as a therapeutic agent. And so here in our strep throat model, if we do the group A strep infection, we have a similar colonization as before. But if we try and treat it with Strep salivarius, we have more group A strep than before. So this probiotic is doing the exact opposite of what it's supposed to be doing. And the reason that is because the probiotic is regulating its antibiotic production through these quorum-sensing peptides. And it's closely related enough to group A strep that group A strep can recognize its quorum-sensing peptides as its own. And what group A strep regulates with its quorum-sensing peptide is the same protease, EB, that we've shown that I just explained is important for reducing the inflammation that's allowing virulence during strep throat. And so group A is spying on this probiotic and microbiota member in order to increase its own virulence. And so we can modify and engineer this probiotic strain in a way that group A can't spy on it. And then now we've rendered it into something that's very therapeutic against these sorts of infections. And so this last example is going to go back and forth between microbe and host to kind of put more together these examples of molecular tox postulates on how we understand what parts of a microbe cause disease and what parts of the immune system are important for fighting that disease. And this starts with another substrate of caspase-1, which I'll remind you is the regulator of the cytokine IL-1 beta. And another substrate of it is called gaspermin D, which is an effector of inflammatory cell death called pyroptosis. And how that works is these gaspermins reside within human cells and they're inert. But when they're cleaved by caspase-1 or other proteins, they liberate an N-terminal fragment. And this N-terminal fragment contains charged residues that will bind the acidic phospholipids in the cell membranes, and also oligomerize into a pore, and that leads to the cell dying. And so this is a really important defense mechanism against viruses and intracellular pathogens that try and hide out inside cells, where they would be protected against immune effectors and antibiotics. And so there's a number of these gaspermin proteins that are regulated by lots of different human proteases. The exception being gaspermin A, which has no endogenous regulator and is also unusual because it's expressed in the skin, which is where group A strep infects. And we found that that SPB, which cleaves IL-1 beta, will also cleave gasdermin A, liberating an N-terminal fragment that looks to be very similar to the sort of fragment that's released by caspase-1 cleavage of other gasdermins. And if it cleaves within this green loop here, and if we engineer a form of that protein without that loop, then SPB won't cleave gasdermin A at that site. And if we force cells to express these different forms of gasdermin A, cells that are expressing normal gasdermin A die, but only when SPB is introduced into the cell. And cells that are expressing this mutant form of gasdermin A, missing that loop where SPB cleaves, so it can't be activated by SPB, they don't die as readily. And then we can more fully characterize what's happening to this gasdermin A protein. And so I mentioned that these form pores in the membrane, and that's how it kills the cell. So it has to bind lipids. Here we spotted different lipids onto a membrane, and we found that gasdermin A somewhat bound these acidic phospholipids here. But if it was cleaved by gasdermin or cleaved by SPB, it bound them much more readily, as we would expect. And if we incubate gasdermin A with lipids, the lipid composition of the inner leaflet of the human cell membrane, we find that uncleaved gasdermin A stays in the soluble fraction and doesn't interact with lipids. Gasdermin A cleaved by SPB, the N-terminal pore-forming fragment will insert to the lipids, while the C-terminal fragment and also uncleaved form stay behind in the soluble fraction. So it is gaining activity as a result of SPB cleavage. It gains the ability to associate with lipids. And we can test this further by seeing if it will punch a hole in lipids. And the way we do this is with a fluorescent dye called sulfurrhodamine, and we can pack it within liposomes at a high molar concentration. And at this high concentration, it's quenched and not fluorescent. But if we punch a hole in the liposome and it leaks out, it becomes dilute enough that it gains fluorescence, which is what we measure on the y-axis here. So SPB and gasdermin A alone won't lyse these liposomes and release this dye. Co-incubated, they will. And this is control of Triton, a detergent, to disrupt the lysosome, so it's a positive control. And so last part of the molecular part of these postulates is to see if it's, again, necessary and sufficient. So here we took human primary keratinocytes from neonatal foreskins, cultured them in vitro, and infected them with our different strains of group A strep. Uninfected, this is a Western blot, and the uninfected keratinocytes have gasdermin A in this large molecular weight form. Infected with wild-type group A strep, you get this N-terminal cleavage product, the same that we see with the purified proteins. If we engineer group A strep to be mutant for this gene, SPB, they no longer cleave that fragment during infection, and then we can complement that gene back on a plasmid. And this is off a really strong promoter, so it's expressed to a much higher level than natural. And it will generate that N-terminal fragment again. So that's fulfilling both parts of those postulates. So finally, to see if that's important, we have to test this in vitro or in vivo in an infection model. And so if we just compare the wild type to the SPB mutant bacteria, we would be able to discover whether SPB is important or not. But we wouldn't be able to know whether SPB cleavage specifically of gasdermin A is important. And so to do that, we also have to manipulate gasdermin A. And the way we did that is by making a knockout mouse using CRISPR technology. One thing that's different is in the mouse that there's 3 tandem copies of the gasdermin A gene rather than just the one that's in the humans. Often, especially in immune genes, this is evidence of evolutionary diversification, because when you have these gene duplications, you can hold one gene to maintain function, while the additional genes are free to evolve new functions. But we went ahead and deleted all 3, so it would be similar to just a gasdermin A knockout human. Gas termin, and so SPB cleaved, just as it cleaves human gas termin on this side, it'll cleave mouse gas termin 1 and 3, and to a lesser extent gas termin 2. However, this gas termin 2, A2, isn't expressed in the skin anyway. It's only the 1 and the 3 that are expressed. So knocking out the whole locus would be the same as knocking out just the 1 gas termin A that's present in humans. And so if we make the mice and then we purify keratinocytes out of the mice and then infect them in vitro, we see that group A strep will cause lysis of these keratinocytes when it's infecting them. And this is SPB dependent. So if you delete SPB, group A won't cause lysis of these skin cells. And the cells from the knockout mice are completely resistant to this lysis, suggesting that it is both gas termin A dependent and SPB dependent. And we see the same thing in human cells where we can knock out gas termin A using CRISPR delivered by lentiviruses, and we see the same sort of phenotypes. And if we then do the infection of the mice proper and see what happens, we find that gas termin knockout mice have much greater bacterial burden than wild type mice, suggesting that gas termin A is really important for detecting group A strep. However, if the strep doesn't have SPB, gas termin A no longer matters anymore. There's fewer bacteria here because SPB is important for the bacteria to do other things. And so, like with IL-1, I had suggested that there's a good part and a bad part for group A strep. It's bad when it activates it in the skin, but it's good when it activates it in the throat because it promotes its growth and transmission. The same thing might be true for activation of gas termin A in the skin. So in this necrotizing fasciitis model, I showed that gas termin A was important for preventing too much growth when it's causing these really severe infections. But maybe that's a good thing for group A strep. It doesn't necessarily want to kill its host. It wants to grow some and get transmitted. And I think it might also promote its transmission also. So this is a video of keratinocytes infected with a lot of group A strep, and just so you could see it a little better here. But you can see as the keratinocytes are dying, well, some video quality here. As they're dying, though, they round up. And as they round up, all the bacteria that were in and on those cells in the color blue, they stay with it. And so we think this is a way that in superficial infections might protect from allowing the bacteria to get too much deeper into the skin, which could be good for the bacteria again, so that it doesn't kill the host. Also, if the video went longer, you'd see that these cells lift up from the Petri plate. And so that might promote the transmission of strep during a natural infection as these layers of skin desquamate, they're shed off. And that the infectious unit for these skin infections aren't free-living bacteria, but these biofilms of bacteria that are riding these dead cells onto their next host. And so that's a broad survey for the sorts of work that people in the molecular microbial pathogenesis field do, and my particular takes on it that I hope will be useful in the future to better understand these diseases and come up with treatments to better treat infection, but then also limit transmission and other issues we have with these diseases. Yeah.
I have two questions. One, in that video, the black, when you play the video, like you see those black squiggly things, that's the bacteria, not the purple, the black thing.
The black things are the bacteria, and some of the bacteria are stained blue also, but all the little squiggly things are bacteria. And so strep are streptococci are balls, and they grow in chains. And so what you're seeing with the squiggly things are the chains of the bacteria.
I saw these squiggly things in my cells. I put them away. They just, okay. Oh, my God, they were contaminated, sorry. for the word. Second question, the IO1 inhibitor drug, is it normally the RAR? What is it?
Anakinra?
Yes,
Yeah.
is that a lifelong drug?
No, so these different drugs, there's at least 5, maybe 6 drugs that are in that product space. A lot of them work a little differently. So some are antibodies, and those typically are administered every month. Some are recombinant proteins that are receptor antagonists, and some are dummy receptors that kind of sequester the free cytokine. So there's lots of drugs in the space. They all work similarly to prevent IL signaling. And they're not lifelong, depending on their half-life. Some are, daily, weekly, monthly.
And, so when, in the study where we looked at the patients and basically the ones that were on this drug and then you looked at the infection susceptibility, was there like a sub-analysis where you looked at how long the patients were exposed to this treatment or like?
I didn't have access to that data, so I was able to look into medical records, and I just didn't get enough depth from those in order to do a good analysis.
Thank you.
There's a question online from Dr. Tiravanzium.
Hey, Chris, thank you for the talk. It was really illuminating on a lot of different subjects. I have a question for you with regards to what you were saying very early in the talk when you were talking about plague transmission and fleas having to pick up the pathogen from the circulation, and then the small blood meal and making sure that the pathogen prevents sepsis while not preventing bacteremia. What's happening in the natural reservoirs for plague? Do you think some of these animals, some of these small mammals, burrowing mammals, actually carry a high bacterial burden in the blood without dying? Because we know for malaria, for example, some adapted malaria, let's say, for example, Plasmodium inui does that in orangutans. They can walk around with very high parasite loads without showing any disease. So is it possible that maybe some of the reservoirs would tolerate high bacteremia with pestis, or is it not in the books?
No, thanks, Raveen. Yeah, I think it's known to be variable, but it hasn't been studied deeply. So in the ground squirrels in the western U.S. where it's endemic, they do die from it. And I don't know the time to death, but they can tolerate it for a good chunk of time. I recall a report suggesting that giant Mongolian gerbils, which are the reservoir in Western and Central Asia, they can have it for years, have bacteremia for years. I don't recall the citation, but it feels like there's some truthiness to it.
Okay, thank you.
Thanks so much. I really love the plane analogy, and I also just like the specificity by which you can engineer and reverse engineer both your pathogens and your host is really amazing. And so I guess my question is a little bit broader. So when I was a PhD student, I studied virulence, evolution, and I looked at malaria parasites, and I got really confused at the time because everyone defines virulence in different ways. And I quickly discovered that virulence factors are a big thing in, people studying bacteria and bacterial diseases. So I wondered, how can you translate some of these? Do you think it's this kind of work that you do, how easily is that translatable to viruses or protozoans or worms? Mm-hmm. And how far do you think we are with that work?
Yeah, no, I think it's the same thing for all of them. So it's just like, a human. Success as a human is having children and passing on your genes, right? I don't define success like that, and I think most of us don't. But evolutionarily, throughout time, that's success. And so what most pathogens want to do is they have been selected for, over generations. The ones that have the most progeny are the ones that have taken over the population and survived. So you have transmission and you have growth. Most of what we think of as virulence factors, I think there's a tendency to go after the ones that have the biggest phenotypes or the most toxic or things like that. But the ones that are most advantageous are probably the ones that are more nuanced at promoting these things. I think there's definitely pathogens that are exceptions. So Bacillus anthracis is a great example where it wants to kill its host. It needs to kill its host, right? Because it is disseminated by spores that form within the carcass of a dead animal, such as a cattle, and so it decomposes, and you walk by and you inhale it, and that's its transmission cycle. So if it doesn't kill its host, it's a failure. But that's really rare. Most pathogens don't want to do that. So it's really just about how am I growing and how am I finding my new host.
Questions online?
I think we're good. All right.
Thanks.